Log on, tune in, find out. Another good idea from Cambridge.
Our next talk is going to be by Marina Savelieva from Novartis. And we are looking forward to a wonderful story on dose escalation and PKPD. Please, Marina.
Thank you, Peter. And first of all, thank you also for inviting me here. For me, it's a very special occasion to come to Isaac Newton Institute, because being a very small child growing up in Russia in a family of physicists who decided it's never too early to start with a scientific education. So with about, I think I was 5 when I first heard about Isaac Newton, and then from then on about all the scientific principles that is kind of modern science is based upon. And okay, despite this very intensive training I undergone in Russia, unfortunately for me, maybe fortunately for science, I haven't done an academic career. So what I'm doing now, I'm working in Novartis in a group that's called Modeling Simulation Group. And actually what we are doing there, so it's the same company, but the scope of the problem we are handling is, or the methodology we are using rather, is very different from what previous speaker presented, so you will see it right away. What I want to talk about today is how the PKPD modeling, so we heard about the term already at least, could be used in optimizing the dose escalation trials in oncology. And this will be much less about the trial design per se, because we are not really involved in trial design, so how do you design the study, but rather about how can we inform the statistician, the clinical team, to take better decisions about the dose escalation steps, or how we can do better dose finding, for example, for the next phase study, with an application in oncology. And this is the topic essentially that I started to look into when I joined Novartis, and more been working in an early phase of development. So this is the outline of my talk. I go through motivation. I will just mention, so basically I don't have such a hard job to do today, because we had a wonderful presentation through this whole week, so the whole job has already been done for me essentially. I just kind of summarizing this a little bit. And then I will show you, I will tell you a bit about PKPD and how can we use it to do the decision making in phase 1 oncology trials. And I will conclude with some concrete examples. Here it was hard for me when I realized that all these talks will be recorded. I had to retrieve all the data. So I will show you the modeling in a very general terms without showing real data. So you have to excuse me for try to keep it very general, because all the trials I'll be talking about are ongoing. It's really work in progress. And then I will end up with a conclusion. So what is the motivation? So as our mission in Novartis, how we see it, what we are doing is to get the dose right. So what does it mean? So it's all nice wording. So it's concretely meaning that we want to have want to be able to take informed decision about, for example, dose regimen for a future trial, as well as, for example, during dose escalation trial, to decide in an informed way, taking the whole information available into consideration about the next dose cohort. And so we talk a lot about the trial designs, and the majority of them based on the binary safety efficacy endpoints. And those are, of course, very often simplification of underlying longitudinal process, very complex biological process that's going on. So usually we consider a DLT event when a safety biomarker, for example, crosses some threshold, and the same for efficacy. So this is exactly the part that we step in with our modeling exercise. We want to explore the time, the dynamical aspect of the process, not just to relate dose directly to the response, that is 0-1, but to see what happens in between. And I think it's really very important. Other topic is about how can we stratify by subgroups, because of course when you come into phase 1 trial, everybody knows this, especially in oncology, it's a highly heterogeneous patient subgroups that sometimes you can stratify using covariates, sometimes you really don't know, you can use some clustering approach, but what is for sure that we can't use just 0 and 1 responses there. So those type of responses, they are not having enough information to do the subgroup stratification, especially in early phase when you have very few patients recruited. To the contrary, if you work with longitudinal data, you can really do this. And different topic here in the bullet point 4, that the processes underlying the dose-limiting toxicities in oncology especially are very often reversible. And so again, this is a dynamical feature. So when you retrieve the patient from the treatment, so you don't give the drug any longer, you hopefully come back to baseline that is acceptable in terms of toxicity. So, and the question is how fast, what could be an alternative treatment, and so how do we manage to best these toxicity events? So just a repetition of what Beat presented in his very nice talk last Wednesday. So what are these phase 1 oncology trials are aiming to do is to determine the maximum tolerated dose. So the idea is we want to be toxic, of course, because we want to fight the cancer, but we shouldn't be too toxic. We don't want to have this, so we have to put some threshold on the toxicity. Where this threshold lies is not always very clear, but so the ultimate goal is to find this MTD. And the methodology we're using now by Novartis was described in the talk of Beat. It also presented is based on this paper of 2008. And I think it's a big success story of Oncology Novartis trial is that they are based now this, instead of doing 3+3 trials, we do this based on adaptive design. And the model is very simple, so it's just based on logistic regression, 2 parameters, and there are some modification of this. You could add some covariates, etc., etc. And Beat also was talking about how we deal, for example, with a Japanese trial when we work with a mixture prior. But I will come to this later on. So what's going on? So this methodology, this is the basis. And what's going on statistically is that after each patient cohort, the information about the posterior distribution of probabilities of toxicities is derived and discussed upon with clinicians, with other members of the clinical team, and then the dose recommendations are given from the trial design, from the statistical trial, that are based on maximizing the probability of a true underlying DLT rate to be in the target interval of the acceptable toxicity, and we have this overdose control here, so we don't want it to be too toxic. So we don't want the true DLT rate exceed this threshold, magic threshold of 33%, so the probability of exceeding this shouldn't be higher than 25%. Good. And the decision is based then on this information, but there are also other sources of information then taken into account. So the clinicians, statisticians, modelers, biomarker people come together and discuss. So where do we go based on the information on their outcomes of our posterior distribution? So where do we go now? Should we escalate? Should we recruit more people in the present cohort? And what is important here, so the part that is considered and discussed is also PKPD data. And this is exactly where we step in and try to do our best to help the team to take good decision about next cohort. Good. So how do these PK data look like, and what exactly do we measure? So here just a snapshot for one patient on a trial where we measure the response versus time. And you see here, so in blue, this was the dose given, and then there was a continuous, so one dose and a continuous dose after some time. And in black, the PK profile was measured, the safety biomarker profile was measured, and also some binary response were measured here it's like 0 and 1. Okay, as I mentioned already before, so safety, so DLT threshold is put somewhere, and what you essentially reduce this, your information to, is to be 1 when these measurements are above the threshold, or 0 if it's not the case for this patient. And essentially what we try to do, which we want to model, because you see what's going on here, it's pretty dynamic. So you need the exposure, the concentration to go pretty high, so there is some delay in the safety response, so it will go up and you have a DLT event. So you certainly need to account for this. Also what's going on here, so you don't give only one dose, you give several doses, and when you withdraw the treatment here, you stop your treatment, you see that the safety biomarker so goes down. So you want to see how fast does it go down, and how can you play probably with dose level and dose regimens in order to avoid these high peaks, because you don't have anything and suddenly it goes high up and then also falls down. Good.
I have a question for
Yeah,
clarification.
yeah.
So on your time scale,
Yeah.
for example, for PK measurements, that may be 24-hour. For PDA, maybe days. How do you put them together? Because it's kind of
I don't
don't understand.
No, it's the same scale here overall.
So, but PK, you are taking blood in patient, and what you are repeating them.
It's repeated measurements, yeah, repeated measurements following the protocol. So here the first profile, it's like, I think it's the first week, and then after several days, and then again, and again, and again. And the same is done maybe on daily basis. So at the same time scale.
So these dots represent area under the curve of PK in each time, or maximum concentration, or?
No, this is actual measurements. It's
But
not.
what you are measuring is AUC in
Plasma concentration of the drug.
at what day? One day.
No, it's a pre-specified by time by protocol. No, it's not IEC. So what you're talking about is non-compartmental analysis. You don't reduce your knowledge. You take into account this is trough. No, not only trough.
No, but at the end it's troughs, that's why.
There is trough concentration. Here are some food profiles all the whole day. So you give it. Okay, good. Maybe I use it like board. That's a good idea. Let us say we give the dose here, time 1,0, and then the concentration goes up. So we take the measurement, and then it goes somewhere down, then you get another one, and then it goes again up again. So you take the actual measurements of the concentration of the drug. And this
8
is
o'clock in the morning, you measure the concentration in the drug, and then one day later, you again measure at 8 o'clock in the morning.
It depends on the protocol. Here, the full, this is the whole week of measurements. It could be also dependent on the half-life of the drug. This is now the story when do you want to measure. So you can talk about optimal PK study design. We're not talking about this. But here we want to assure that we can estimate, so these measurements are done in a way we can ensure that we can estimate half-life of the drug and time steady state. So estimate the also absorption elimination phases of the drug. So I'm not going to talk about this, but what you're saying essentially is also very important, because you have to be sure about that these repeated measurements here, they have to be, so the patient has to be followed for quite a long time to ensure that we can estimate time to steady state, because you give here repeated doses. So every day, so you start with the first dose, you wait for 1 week because you want to have the full profile of the drug, and then you start with the daily dosing. Okay, and then of course, so these measurements are not happening at the same time as the PD. So this is another point. So if you're doing modeling, you don't need to adjust those. So you don't need the measurements to be at the same time, PK and PD points. Good. So, okay, and the last point here is that what you measure, additionally to what I showed already, are the covariates. So you measure you assess the patient individual characteristic in terms of baseline, so age, sex, race, lab values, etc., etc. So they could be assessed at baseline. This could be continuous covariates for lab values, for example. And then you will try to estimate the impact of those on the profile. Good. So here, just a cartoon to show you. So what Merlin simulation is aimed to do is to understand the causal chain of drug action. So what the trials are doing, we want to estimate what the dose given, how does it relate to response, But of course, it's not a simple process. So it's not just we give the dose and we have a binary response. There is a whole biological process underlying this. So we give the dose, and here I write dose regimen. So it means not only the dose should be taken into account, it should be also dosage, interval, route of administration, delivery system. Then it transfers into the PK. So it provides some signal in the system. So you can measure these PK profiles. You activate the site of action. They provide you with a biosignal. Then you can measure. So you have this biomarker you can measure. And then it will be translated into the response. It could be safety, tolerability, efficacy, early clinical results. And so the whole science, so this part we refer to as pharmacokinetics, this part refers to as pharmacodynamics. So of course it's not a split at once, so this all should be then considered in unified framework, and then it's called PKPD modeling. Okay. So another aspect of to this that of course we're not working with a single patient, so we're targeting our therapy to the whole, hopefully, population group, and all the patients are different. So we have to consider, we have to take this inter-patient variability into account. It's very important to account for all possible sources of variability, and the population PKPD methods are exactly aimed to do this. So here, just to show you, so you give the dose to different patients. So here, just one dose and corresponding PK profile, and you see that there's quite substantial variability in the concentrations. So taking only mean or reduce this information only to mean, so doing some naive pooling here, you can lose a lot if you just do it in a naive way. So how do you go? You describe the system, you describe this dynamical process with a structure of PKPD model. This is essentially, so mathematically speaking, it's just a system of ordinary differential equations. And then you use the population PKPD technique to determine mean variance covariance of PKPD parameters of a given drug for this patient population. I'm not going to talk in details about what does it mean, but just to say you saw nonlinear mixed effects models are exactly used to work on these parameters estimations. And there, so for us, from Novartis perspective, it's very important to have a handy software that we can use. And also when we work with health authorities, so we do some analysis, we should be able to, so FDA are looking on our outputs and should be able to reproduce them. That's why a question of software is also very important. So all of us are mathematicians in the group, so not all of us, part at least, we would be able to write our own codes, unfortunately, so it's not allowed, and also often we don't have time. So we still are restrained to work with some industry standards still. It's non-MEM software. I don't know how many of you are familiar with this, but this is pretty old software, I think from 70s something. And the problem is not even been so old. The problem is that it's based on linearization of the likelihood of the model. So it's not even real maximum likelihood estimation of this first order FOCE methodology that is behind it. This means that you have all possible problems related to this. One of those that I don't particularly like so much is your model just not, it's very difficult to fit the model, it's very sensitive to the your prior belief, so your starting values. And so you, because you know, it can converge to any local minima, so if you have, for example, you don't have very rich data structure, and you have a flat likelihood, you can get to any point and it will say you, okay, this is your estimate. You change your starting value a little bit, it provides you with another one. Of course, this is not exactly what we want to have, especially here. It's often the case in phase 1. We do have a very sparse data set. We don't measure the PK and the PD often enough yet, so we want to have something better. So this is another software that I think quite nice, and we also testing this now in modeling simulation. Some of us are also using this routinely. It's a Monolix that based on a stochastic approximation expectation maximization measure that is already a much better tool. So you can talk to France if you want to know the details. So here the paper reference to. So it's really already nice advanced, very nice advanced compared to this non-MEM. Unfortunately, it's still for us kind of a hurdle to submit our program sometimes to FDA, and also in terms, I don't know if it's now a standalone version. For a long time it was MATLAB based. So for those people who were not familiar with MATLAB, it was not evident, and also the way we work on our Linux cluster machine, so it was not installed there, so it was not so easy. Now, alternative that I'm sorry I prefer is to work with Bayesian methodology. So, because very often you will see you have to estimate your parameters, you want to introduce your prior beliefs, and you want to do predictions. So for me, this is a nice setting. The Bayesian setting is really nice and sometimes most appropriate ones, but software here is a real issue still. So I was trying to work with the Metrum Institute library. They developed an add-on to Winbox. So also it's another story. So do you like Winbox or not? Do you want to do something more fancy than a Git sampler? Sometimes not so efficient. So I tested this software, by the way, and yeah, the results are pretty satisfactory. It's not very easy. So most of the examples I will show you later are done in Bayesian framework. Good. So how does Bayesian population PKPD looks like? So what you have to understand here is just it's a hierarchical Bayesian model. So you have 3 levels of hierarchy. You have your model specifications that are conditional on the individual parameters and residual role variance. So you have to specify the distributions here. You have to specify your model for PK and for PD. You have to specify on the second level distributions for individual PK and PD parameters. So typical choice, it's multivariate normal distributions with mean that is accounts for fixed effects and some variance covariance matrix, and the same for PD part. And then of course you have to specify priors for parameters. This is the whole topic in itself, the way you're doing this. So some choices are inverse gamma, Dirichlet, multivariate normal for mu and C here. And then for variance covariance matrix, it's inverse Wishart priors. So I also investigated this a little bit. And there is a whole discussion, should we do this way or not? But it's not the scope of today's presentation, but just to show you the complexity of the model. And this you are doing this specification. So you have K patients, so the I is the patient number, and J the occasion you measure your PK or PD endpoint. Good. So so why do we want to use this methodology? So I explained this hopefully already a little bit, but just to repeat here. So by using this PKPD method, you can include this time course description into your modeling, and you're doing this via a mechanistic model. So you have a comportamental analysis. Once you have this comportamental model, you can do predictions from it. You include fixed and random effects, so you count for different sources of variability. And also linked to this time course, you can then estimate how alternative dose regimen would impact your safety or efficacy profile, or how would it purely impact your pharmacokinetics profile. And of course, it's not all that easy, because you have, first of all, so your functional relationship. So your model is a solution of system of ordinary differential equations, so you have to have good integrated methods to find a solution, but also it's not always evident so what the model should be. So the model selection is a challenge here. So there is also a branch that works with this. So just because in terms of the comportamental analysis you don't understand what parameters means sometimes. There are some physiologically based PK models that also even more complex. You have a lot of parameters that are referring to different parts of the body. So, and then at least you can measure the concentration or respective biomarkers in these parts. And so this is the way to validate this, but it's, it costs you a lot, because this is a huge, so sometimes you can end up with as many as hundreds differential equations in the end. So we don't really work very often with those. Again, so identifiability issues. You have a lot of parameters, and very often your problem is ill-specified, so it's an ill-defined problem. You hope, so this is from a mathematical perspective, you hope that your data, so there are 2 types of identifiability, mathematical and statistical. So you hope that you statistically speaking, you have enough data to support your model to be able to estimate your parameters. And as I said already before, working in oncology, our data are heterogeneous and very sparse. It's sometimes depending on the methodology you use, it's difficult to fit nonlinear mixed effect model. It's very time consuming, and in Bayesian setting is always a discussion on what prior should be. So how do we integrate prior belief if we don't know anything about the drug yet, and if we can't integrate our prior belief, why should we do so? So it's a separate discussion. So what is our goal? Why do we want to work with? So we want to start doing PKPD in earlier phase compared to what's been done till now. So when I joined Novartis 5 years ago, this was a rule that the whole population PKPD analysis just started when we were about to submit a dossier, I mean in phase 3 already, at the very end. So we're working in this confirmatory kind of modus. And definitely it's too late to make an impact on the decision making regarding the drug development then. So essentially what we're saying all the time, guys, you know, we have really nice findings here. It's pity that you didn't know this before, because otherwise you would be able to integrate. It was not very satisfactory neither for us nor for clinical teams. That's why the discussion started that we have to do it as early as possible. So this is what we're trying to do right now. And our hope is if we can do this early enough, we can avoid treating patients at unnecessarily low high dose. That is still often the case, unfortunately. We can reduce the total number of patients. We can help with dose selection or justification for the future trials. We can detect subpopulation with better responses to the treatment, and we can synthesize the information across the various trials. So we can pull the data all together and analyze them all together. So leverage this knowledge. And the whole, of course, and sums up an optimized drug development process. This is the goal. I look a little bit into the literature, and I found that there were already so 96 already there were, of course, some thinking about how can we integrate the PK, for example, into our trial design. So Piantados and Leo in their paper acknowledge the fact that in comparison of at least PK information into the dose escalation trials is extremely important, and it could improve efficiency and accuracy of the studies. This is specifically the case when we have these oncology phase 1 trials with a very high interpatient variability and a complex drug profile. So what they propose in their paper is just an extension of a CRM methods where they have this 3-parametric model, and they account for a PK part here with this delta AUC. So saying if your AUC is higher than expected, then essentially your probability goes up. Good. So now I'm coming to the concrete examples. How can we and when should this modeling be done to support the decision making? And I give you 5 examples. Again, I'm sorry I can't show you the real data, so it will be more of a description again. So, and those 5 are related to the situation that I just experienced over the past months, where we had on a concrete trial complex, unexpected PK profiles, heterogeneity in the patient population. We wanted to target a specific subgroup of patients. Here it was an ethnicity study. And we wanted to evaluate alternative dose regimen to those who were already tested during the study. And we wanted to justify dose selection for future trials. Good. So the first one is about complex and unexpected PK. So here, this is a phase 1 oncology trial that is ongoing currently. And the knowledge about the PK was such that the estimated half-life of the drug was some hours, and time to steady state of the drug was supposed to be also less than 24 hours. When we started to recruit the patients and we analyzed the first cohort, actually we realized that it's a very strange picture that appears to us, that essentially the half-life is much, much longer. So it's over 30 days, and time to steady state it's almost 3 months. When we communicate this to the team, nobody wanted to believe, because of course the way we are doing this, so we run some animal studies, then we try to scale to the humans, and there were some consideration about the long half-life of the drug, but not to this extent. So in the beginning, nobody wanted to believe us, and the reason was that the data were collected also in the beginning of the trial up to here, and also the naive pooled analysis didn't provide the same estimates. Okay, we insisted a little bit, so we said to the team, please augment your protocol. And the first version I think was about following out the patient for to detect agility for cycle 1. Cycle 1 being 28 days, you understand pretty fast. So if you stop here, you're not very well done, because your concentration is still going up, and potentially you will detect DLT later on. So what we said to the team, please augment the protocol. They did it, included this follow-up for the patient for at least 3 cycles, and also more frequent monitoring of DLT events. This resulted in different, much lower MTD dose that was expected in the beginning. So this was a very nice impact, and the program is still ongoing, but so it's not clear why the drug having this weird, what's going on here, what is absorption, elimination, but at least we were able to make an impact here. Okay, so the second example is about the heterogeneous patients population, and I will talk about the safety endpoint, myelosuppression related toxicity event. This is also very common in oncology. So it's a concrete, also again, it's a concrete phase 1/2 trial, so it's a joint trial here, and myelosuppression was detected to be a main DLT event. So myelosuppression means just the number of blood cells just drops below a certain threshold. And standard analysis tried to examine the relationship of probability of getting DLT versus dose or an EUC, and all of them failed, because when we look at this, it was very weird. Higher the dose, lower the probability of getting DLT, higher the EUC, lower the probability of getting a DLT. So it was not really satisfactory to the team. They came to us and said, okay, what could we do about this? It's very strange. What we did, or I did concretely, I fitted the indirect response PKPD model. So I will show you the model here, so you understand a little bit better. So it's a 3-compartmental model here for the PK part. So you give the dose, it goes into the central compartment, then redistributed somewhere, and then it's eliminated from the central compartment. So this all described by the system of, it's first order kinetics, so it's a system of 4 differential equations with initial conditions, and you want to put an effect somewhere. So here I found out that the best model, the best fit provided me when I put the effect compartment on this peripheral one, and you say, so you PD, so this is the equation describing your essentially, you PD the production of the blood cells. So the effect here is on the 0 order production rate. So when the drug modifies the production rate, it inhibits the production rate. So when you give the drug, the production rate goes down. And we suppose that at time 0 we're in steady state here. Okay, back to my previous slide. The findings were the following. So when we look at the time course of the blood cells, we saw that you give a patient a dose, the number of cells goes down to a certain extent and then goes back again. And then we examined also the impact of the covariates, and this is where we really were surprised to see to what extent, so not surprised to see, of course, because pretty evident afterwards, but baseline plays a major role. Of course, if you start very low, you have already the previous treatment, and you have already the myelosuppression, then the next dose will just kill you. So you go immediately below the threshold, whereas if you are doing pretty well in terms of your baseline values for this number of blood cells, you can get pretty high load of the drug and you will still be doing all right. So, and also what we detected additional to this, that you can differentiate by tumor types. So different tumor types will have different profiles. So I will show you what we simulated for the clinical team to see what's going on. So here tumor type 1, tumor type 2, they are starting also, they were differentiated also by baseline value, so it's the baseline value for typical, so they are typical patient profiles, 140 here, 250, a little bit above. So and we simulated different profiles for different dose levels. So you see, so you get the dose, it goes down, get the next one further down, then you don't get the dose, so you're going up, then you get another one, etc. So you see this nice depends between the dose given and you can estimate the time you need to go to fall below this threshold, but you can even more importantly estimate the time you need to recover. So you can play with this a little bit in the clinic and to design concrete patients. So how do you monitor? If you see that the patient approaching this threshold, you can withdraw from the treatment and then your blood cell will go up. Okay, and so this is exactly what I already mentioned here. So this goes into the selection, my third point, how we can be useful with our modeling. So for evaluation of alternative dose regimen, here the team asked us to compare two regimens. So it was weekly regimen, so the dose given weekly versus every other week. And we wanted to have better safety profile while keeping the same efficacy. We don't know yet, of course, here what the efficacy is, but we hope that at least keeping the same level of exposure, similar level of exposure should provide us with a similar efficacy. So here, exactly what we simulated. So the two profiles, exposure is the same, doses are different and different dose regimens. So here we give the dose 3 times weekly, then we stop for the whole week, then do it again, and we see we don't run into troubles here. Whereas for the same patient, it's a typical patient here simulation, we go pretty fast. So if we do it really on a weekly basis, the same exposure provides us with a more of a problem. So we get into DLT then. Okay, so subgroup stratification. This is an important one I want to show you. So Beat talked to you about a very nice Bayesian approach and how they tackle this with the mixture priors. So what was important here that we looked into this more precisely, how we can add something to this, and it was important for Novartis in general. So this Japanese question coming all over again, because essentially what's going on, so the Japanese are very particular about, you know, developing drug for their specific population, and this is ethnicity could be really a factor impacting their response in these patient subgroups. So it's very cost-consuming, time-consuming to run the trials in Japan. We would like optimally to run just a separate arm there, but for this we want to show, we have to show to the health authorities in Japan that we do have the methodology for this. So what the thinking we were given to this, how could we use the model to optimize the whole process? So we could use to select the prior distribution for the more based on model-based design. We could help with a better starting dose. But finally what we did now is a small piece at the reassessment of the probability based on the PK parameters estimation that were coming from the population PK model. And what we did concretely that we were observing in an ongoing Japanese trial the DLT events, but the sample size was very low. So it was not really particularly clear, is it just by chance or it's due to some systematic difference between Japanese and Western populations. So what we did the pop PK analysis with the Japan as a covariate, and we did find significant differences between two populations in terms of their pharmacokinetic parameters, and the PKPD model we fitted afterwards. So we showed that DLT are driven by peak concentration, having very large differences in the PK, will get high probability in Japanese population of getting a DLT event at the same dose level compared to Westerns. And this seems to confirm now. So the trial is ongoing, and we really see more and more of the Japanese getting, at the lower doses, getting DLT events. So probably we have to go into lower doses as it was foreseen till now. Okay, so I think I have how many? 1 minute?
5 minutes,
Still?
short 5 minutes.
Short 5, okay, good.
Very short 5 minutes.
Okay, this is another thing that, you know, I could skip this one just to say you that, you know, this is something that also FDA challenges us sometimes while we are in phase 1, so how can we justify already something that we are planning for phase 2 regarding the efficacy? You don't know what the efficacy yet is, so we have to do some simulations on some, and meeting some very strong assumption to see. So here in this trial we were considering 2 doses, so to show there is real differentiation between 2 groups, and essentially it was very welcome. So the FDA liked our simulations. We simulated the inhibition rate of a certain biomarker, so, and we showed that, you know, in percentage of population, so per dose here level, we showed that 1 dose will really, especially for the high levels of inhibition, where we say this should be really where we maybe could relate this to efficacy. At the high levels, we see the differentiation between 2 doses. Good. So I'd like to conclude now. So what I wanted to show you that we advocate in favor of doing the applying population PK methodology early in clinical drug development. And we exploit, so this is still challenge, and I have to say even in the group, so not everybody is agreed that we should do it on a regular basis, but when we can show that we really provide an added value, what we call all the time, we have to provide an added value. So these cases are kind of, justification why should we do this. And so these concrete examples here are very important. And based on the experience so far, and I hope I made this in my presentation, I try to explain you why the description of the time course is so important, and how this transfers into the predictions we are able to do about the different dose regimens and inclusion of the covariates. And we do this analysis, of course, it's adaptive in a sense that as new data become available, we integrate them into the analysis. This is also why I like Bayesian methodology so much. So you don't need to run the whole model, you just update your prior information. And you have essentially your posterior predictors. So you have to simulate a lot. So you have your posterior predictive distributions ready to hand. So it's very easy to do to provide a team with predictions based on those. And of course, modeling can be challenged still. It's a very complex model that we use here. And we don't know so much about the drug yet, so it's even more complex. Data varies fast. It involves a lot of assumptions. So what we are doing now, because sometimes the team forgets about what we were saying in the beginning, what assumptions were. So please do not some mention. We don't know anything about the drug, but you could do something for sure. So it's really we have to explain. We do have some limitations here. We do a lot of simulation, and so modeling is not the most complex part. So you have to understand how can you model, how you can select your model and error structure most importantly. But simulations then is what's really time consuming. We have the timing issues. We have to be fast. So in order all this to be accepted, we have these dose escalation meetings like sometimes in a manner of 2, 3, 4 days. We have to provide already the new simulation adjusted for new data that just became available. And of course, sitting alone and being a modeler and doing online modeling wouldn't bring anything if we wouldn't be able to communicate, to collaborate with other team members. So we have to really explain the methodology to get it accepted by clinicians, by statisticians, clean farm people, biomarker people, etc. So this is also essential part of our job, the communication here. And I'd like to stop here, and thank you very much.
Wonderful talk. Any reactions, comments, Basha?
Yes, I just wonder because what you are doing is to put the information of PKPD into your dose regimen, but you don't optimize the times for PKPD.
No, it's not about optimal PKPD.
Yes,
so
I know. But would you think that would be possible to add into your models?
So it depends on the trial. When we talk about, so this is not a particular trial for PK, so there are separate trials where we have to do this.
The measurements have to be taken anyway.
Yeah. No, but we consult the teams when they write the protocols also in this regard. So we want, of course, be able to estimate all the phases of the drug, so absorption, elimination, and distribution. So yes, we do this. But this is slightly different topic. What I wanted to show today, just including this time component, this dynamic component.
Have you tried to optimize times?
Yeah, we're doing this as well. Yes, of course. But this is, it's not a phase 1 trial, so it's more of a work for phase 2 trial then that we're doing this.
So maybe that's a good opportunity for a retrospective advertisement. The talks are all recorded, so you can actually go back to previous talks. Basha gave a talk on Monday afternoon talking about optimization, like optimal choice of the sampling times in this context. So you could come to the talk that has already happened. Yeah, they're actually pretty good. All these talks are online, beautifully synchronized. It's entertaining. Yeah. Sorry, Jack was first.
Yeah. So for the PK and PD part, do you put these two kind of independently?
Yeah, it's a very interesting question. So this is something I explored as well. How do you do this? So sequentially, or you feed all the data at the same time, and of course with all advantages and disadvantages. So you do want to propagate uncertainty from PK into the PD, and so if you do it in a Bayesian framework, you don't have so much to worry about this. If you do it in a frequentist framework, you have to give some thinking into this. Practically speaking, we first collected, so PK data first become available. So traditionally, we do PK separately and then plug in, so hopefully, so individual parameter estimates into the PD.
Oh, wait a minute, I thought it was an indirect response model. I thought it was one model for both.
No, but you can do this separately as well. So
Okay.
first you feed your pop PK, you know. It's also very challenging to feed the full PKPD, you know, because you have to really be sure that
But
there are
then-
so many possible combinations on the PK and PD side. It's too complex sometimes. Yeah. So-
I don't work in this area, so I don't claim to be all that knowledgeable, but in your data, you seem to be measuring white blood count and the concentration of the drug in the blood over time. Is that right? So you have the two longitudinal processes.
Exactly.
Did you model those as a bivariate longitudinal normal?
Those were the ODEs, no?
The ODE is also a differential equation. So this is solution. You say that
That is, how did you get at the association between those two and when you finally got down to the model?
here, you know, you say that this is a description of your time course, the solution of this system of differential equations.
Yes, so you've got the different amounts in the cells, but you've got the agent in the modules that you've got the agent in the blood, but there's also the white blood cell count per unit volume in the patient's circulatory system.
So here,
is that what
here
R
you
is?
measure this. You measure this. R.
That's R.
It's
So
R.
what did that look like in terms of the statistical model?
The statistical model.
So it's comparable, non-linear mixed
Equation.
effect. No, non-linear mixed effect model is a constant for this.
It's a mathematical.
This is a structural model to describe the process, but
Yes,
then we
I
constant.
understand. So what did the statistical
No,
model come out
The
as?
statistical model is what we are. Okay.
That's a statistical model. This one.
Is
Yes,
there,
that's
there we
It's
go.
statistical,
So
yeah.
everything was multivariate normal?
Yes.
I see.
Yes.
Inverse,
It seems to me that
inverse
the the Bayesian non-parametric methodology for this is quite well developed, isn't it, Peter? I mean, Gary Rosner has done an awful lot of work in it.
It's only variations on the priors, like instead of the multivariate normal.
Yes.
That's probably.
Yeah, but you know, it's not always multivariate normal. So I took the very general case here. It's not always the
If it's
case.
Bayesian, it's very simple.
But it's very simple, you know. It's a simple. Yeah, it's very simple. You know, I mean, this is already complex enough for me. I have to say, I'm not
It's almost
always work with
free.
multivariate normal distributions.
And any more comments, Basha?
A little bit different thing. You've mentioned at some point that when the blood cells, the blood cells drop down too much, the patient is withdrawn from the study.
No, I haven't mentioned this.
No?
I haven't mentioned this. Not in all the studies. Not necessarily.
I see.
No, not always. It could be the case, or at least, you know, it depended on the trial. It could be withdrawn, not withdrawn. But we, you know, we recommended to follow up the patient to know. Even if it's withdrawn, we follow up. What the question is exactly?
Well, I just wondered if it would be possible to complicate your model a little bit more and give this patient some remedy to push up the white blood cells numbers.
But they're coming back, you know, if you withdraw from the study, they're coming back, so you don't really need to give any remedy.
Well,
Or you want to
no, what I meant to say is that if you notice that these toxicities are too strong in general,
Yeah.
you could try to remedy this somehow and treat the patient again after some period of time.
yeah, but this
So-
is what you go, you just don't give the drug anymore, and then it comes back to normal.
I assume they have to follow the protocol, right?
Yeah, yeah.
But we should
Maybe
probably
we should continue.
move on to the next talk. So let's thank Marina again for a wonderful talk.